Good. It's 12 noon GMT. A very good afternoon from Accra, Ghana. I hope we are all well. I can see that 31 people have signed in. We hope a lot more will join us. I would like to call this meeting to order. A very good afternoon from Accra. Everyone signed in. I can see a number of familiar faces. Thank you for joining the African Plant Breeders Association in this maiden seminar for APBA seminar series. I would like to first thank the Communication and Visibility Subcommittee of the Executive Committee of the APBA for working hard over the past few months to put together a concept that is working us closely to our next conference to be hosted at UM6P, Morocco in October 23 to 27, 2023. We are working hard to ensure that our third conference is perhaps one of the biggest ever. I would like to thank all those who have worked hard. In fact, quite recently in January, we had an excellent workshop at the Plant and Animal Genomes workshop that we held, which was the first of its kind by the African Plant Breeders Association. I would also like to inform members of the African Plant Breeders Association that the executive committee has been working around the clock. So far, we have constituted 6 subcommittees which are working hard to ensure that we, of course, meet all expectations, so we are able to roll out strategies which allow us to come together and work as plant breeders working for the future of food and nutrition security in Africa. For today, I am excited to introduce the chairperson of the Communication and Visibility Subcommittee of the Executive Committee of the APBA. He's in the person of Mr. Dean Mungani. Dean is a former president of the Zimbabwe Plant Breeders Association and the current product manager green crops of the International Institute of Tropical Agriculture, popularly known as the IITA. Dean Mungani will be introducing the speaker for today. Before he introduces the speaker for today, let me thank you, Dean, for the hard work that your committee is doing on behalf of the African Plant Breeders Association. We believe that it's going to be non-stop between now and October. Every month we would have an APBA webinar. We want to attract the best in the world to speak to us so that we prepare well for the task ahead. So without further ado, Dean Mungani, former president of the Zimbabwe Plant Breeders Association and products manager of grain crops, IITA, may I invite you to take the floor. Dean, thank you very much.
Thank you so much, Prof. Welcome everyone. Good morning, good afternoon, good evening, wherever you're joining us from. I hope I'm very loud and clear. So this is our first webinar. In 2023, we are planning a webinar the first Wednesday of each month from now up until we get to the conference in October. So our speaker today is Dr. Leena Tripathi. She's the director of the East African Hub and the leader of the biotechnology program at the International Institute of Tropical Agriculture, popularly known as IITA. She's leading the transgenic and gene editing research at IITA. She has been involved in plant biotechnology research for more than 25 years with specific interests in crop improvement. She focuses on science to practice and linking scientific innovations to practical applications to solve food production issues worldwide. Her primary research focus on genetically improving important staple food crops like banana, plantain, cassava, yam, to control diseases and pests. Her scientific contributions have been recognized internationally through several awards and honors, such as Excellence Awards for Outstanding Scientists and Publications. She has been honored as an elected fellow of the American Association for the Advancement of Science for her contributions to agriculture. Dr. Tripathi and the team have established a robust genetic transformation platform at IITA Kenya to develop transgenic and gene-edited products and transfer these technologies to national agricultural research systems in sub-Saharan Africa. She serves as an editorial board member for the Plant Biotechnology Journal, Communication Journal, Frontiers in Genomic Editing, Scientific Reports, and Plant Cell Reports. She obtained a PhD in plant molecular biology and MSc in molecular biology and biotechnology. She worked at the University of North Carolina in Greensboro before joining IITA. May I take this opportunity to welcome Dr. Tripathi to share with us insights. Thank you.
Yeah. Hello, everyone. First, I would like to thank Dean and also Professor Eric Denkwa for inviting me to give this webinar. Today, I'm going to actually talk about the application of gene editing in the improvement of African crops. As you all know that, our major global challenge right now in agriculture is the food security. There are actually billions of people who are hungry and malnourished. And as the world population is projected to reach like about 10 billion by 2050. That means there is more critical need for increasing the agriculture production. But when we come to Africa, actually the population is even projected to double by 2050. So even the challenges is more. In addition to the population, there is another challenge, which is intensifying the food security problem. That's the climate change. Because the climate change has harmful influence, not only on plants, but also on pathogens and pests, which are affecting the crop production. And we already started seeing the negative impact of the climate change in the agriculture. So that's why there is a need for the sustainable intensification, and what is required is actually to close the yield gap in the staple crop, whether that's maize, wheat, or bananas, or cassava, and utilize all the tools which are available in our toolbox, including the new breeding tools such as gene editing, in complementation with conventional technologies to increase the agriculture production. So, with increasing demand for food and with the limited resources, we need better and more efficient ways to produce food. And so that means that we should use all the tools available to us, including the breeding innovation, using the techniques like transgenic and gene editing. So what is gene editing? Gene editing basically is a group of technology, is not only one technique, which provides the scientist the ability to make a small, precise, and heritable change in the genome of the organism. And basically, this is the enzyme which only make that small cut. And then after that, the cell's own DNA repair machinery actually repairs it. And in the process, actually, it creates some mutation. And normally, it lacks any foreign DNA integration in them. So basically, in a layman language, if I'll say, you can visualize this process as you edit a manuscript, you paste, you delete, sometimes you replace, and that's what this technology is all about. Gene editing for the crop improvement is actually not new. Nature has been editing the genome for a very long time, creating the variations. And then after that, the farmers used to select, and that's how the domestication and the improved varieties came in. But in 20th century, these mutations were accelerated using either chemicals or the radiation. So in 1895, there was a discovery of radiation, and in 1927, there was the first radiation-induced mutation in plants were reported, followed by chemical mutation, and then later on, more technology came in for the precise gene editing, including meganucleases, zinc finger nucleases, and then more later talents. And then in 2013, actually, the CRISPR-Cas9 editing was reported in plants. And it didn't stop there. Actually, it's keep on evolving, because then there was base editing, the CRISPR activation, and more recently, the prime editing came in. And CRISPR-Cas9, as all of you know, actually won the Nobel Prize in Chemistry in 2020. So this gene editing, as I explained before, so it's not one technology, it's like a group of technology. So there are different endonucleases which can be used for editing, such as meganucleases, zinc finger nucleases, talents, and the CRISPR-Cas9. But all of them works more or less in a similar fashion. So what happened is they create this double-stranded break. And once this double-stranded break is created, actually, then the cell's own DNA repair machinery actually repairs this break. So the break can be repaired in two different ways. One is the non-homologous end joining. And in this case, when the repair happens, it can create some mutation. That can be a small insertion or deletion. The second way of repair is the homology-directed repair, and that repair happens when there is a repair template is present. And in that case, actually, there is insertion. And so the insertion can be very small insertion, or it can be the insertion of the whole gene, or this can also be used for the allele replacement. So gene editing, actually, you can use it for different things like, knockout of the gene, or even the replacement of the allele. So in terms of all the different tools, actually CRISPR-Cas9 based gene editing, which is actually derived from immune system in bacteria, because bacteria use the CRISPR system to actually defend themselves against the viral infection and as well as bacteriophage. So the CRISPR system from the bacterial immune system was adapted for the plants and animals and even humans. So for this technology, CRISPR-Cas9 has been actually successfully applied in several crops. And among all these technologies available, CRISPR has actually become the most popular genome engineering approach right now because of its feasibility. It's simple to adapt. So this, as I explained before, the CRISPR-Cas9, you can use it for the knockout of the gene. So that means you can disable a gene function. You can also precisely replace the genes or insert the new gene, or you can even use it for activation, upregulation, or the downregulation of the gene as well. So there are several ways you can use this technology. It is fast, it's cheaper, and is very accurate and efficient. And you can even use it for stacking different traits as well. So as I was explaining before, so I can explain more in a pictorial form how you can use. So, so CRISPR-Cas9, you can use it when once there is a double-stranded cut with the repairs. It can actually be deletion, or you can have the insertion of the gene, or you can have also the allele replacement. So that's one way you can use. The second technology, which is more recently developed, is like the CRISPR, where actually the Cas9, which is more act like a scissor to cut, actually you can make the Cas9 as dead, and then this can be used for the activation or the repression of the gene. And in this case, actually the Cas9 binds at the promoter region of the endogenous gene you want to upregulate or downregulate, and then that way you can use this technology. The another one you can use is for the base editing or the prime editing, is where you only want to make like a single change, and in that case there is no double-stranded break, but is the nick, so is only one strand has a nick, and then based on that actually you can change either G to A, or you can change A to G or C to T. So that's like a base editing you can also use for it. So it's very important to understand, when you are doing the gene editing, what different type of products you can get, because that has the implication on the regulatory. So all these endonucleases I talked about, like meganucleases, zinc finger nucleases, talents, or CRISPR, they are all called site-directed nucleases because they are very specific to the site, and then they make the double-stranded break. So that's in the short form, we call them SDN. And then based on the type of the repair, you can end up with 3 different types of products. So when there is a non-homologous end joining, that means there is no donor template there. So, during this, there is a targeted random mutation. And basically this one is SDN1 because, there is no foreign gene integration there. There is only a small deletion or insertion. And this is mainly used for knockout of the gene. That means you are silencing the gene function. The second one is when the donor template is present, that is a homology-directed repair. And in that case, there can be a small insertion. And if the insertion is very small, is called SDN2. So this is mainly the few base pair changes only. Or it can be the full gene insertion. And when there is a full gene insertion, that's the SDN3. Or this is also can be the allele replacement. So most of the countries, the SDN1 type is because the mutations are very similar to the chemical mutagenesis or spontaneous natural mutagenesis, they are not regulated as GMO. Rather, they are treated similar to the conventional varieties. Whereas the SDN3 type, it can be cisgenic if the insertion is from the same plant species, then they are also not regulated as GMOs, but it can be the insertion which can be the foreign gene as well, depending upon the donor template, what the donor template is provided. So if I compare the gene editing like basically with the conventional varieties coming out from the conventional breeding, then the conventional, any product which are coming out from the conventional breeding, actually they are not regulated as GM. But there is no product which are not regulated. Some products are regulated only for the, whenever the breeders produce a new variety, we will not say it's not regulated. There are some regulation before it can be registered for commercialization, but they are not regulated in terms of the biosafety regulation. So that's what I mean when I'm saying the regulated or not regulated here. And then all the varieties which are like genetically modified, they are regulated. But when it comes to the gene editing, the SDN1 are similar to this one. SDN2 also in many countries, actually it is very similar to the conventional breeding, but some countries they are That's why is a different color, because some countries only says that they only SDN1 are not regulated. And then when it comes to the SDN3, it depends, because if there is a foreign gene, then it will be regulated as GM. And if there is no foreign gene integration in them, they are not regulated as GM. So this is the overview of the global legislation of gene editing. So the countries you see shaded in green, these are the countries where the guideline for gene editing is already available. And then according to that, any gene edited crops which has no foreign gene integration in them, they are not regulated as GMO. The countries which are in red, which is actually mainly the Europe, the South Africa, and New Zealand, these are the countries actually where the gene edited products and the GMOs are treated similarly. And the countries which are actually shaded in yellow, those are the countries which are under discussion right now. So if I give you more information from Africa, actually Nigeria and Kenya, they were the first countries actually who developed the guideline for the gene edited product. Then later on, it was followed by Malawi. And then more recently, actually, the guidelines were approved in Ethiopia and Ghana. And then there are several other countries actually which are where there is under discussion. Actually, you will be very surprised to see that, you know, this graph is changing so fast that every few weeks you see the countries has different colors. So if I give you little bit more detail, what happens? So if you have developed a gene edited product, actually it the path which the conventional varieties take. And so that means, like, you know, like the hybrid, so they don't need to go through the biosafety regulation. But if there is a foreign gene integration in them, then actually it will be the similar path as followed as the genetically modified organism. Okay, so again, giving a more detailed example here. So the conventional breeding. So when you do the conventional breeding, you also need, if the trait you are interested in, you need to have the trait in one of your parents, then only you cross and you get the variety. And most of the time, the challenge is the narrow genetic diversity in the conventional breeding. But when you come to the mutational breeding, you can either use the chemical mutagenesis or you can use the radiation. But the major challenge with the mutational breeding is it is very time consuming, and then you have to actually screen a lot, and that's why it becomes quite expensive. Then is the transgenic. So if you use the transgenic, that means the genes are always coming from the other species. And then there is the regulatory complexity, but also the cost is a little bit more. But then this is actually, in terms of the time, it takes less time in comparison to these two. But when it comes to the genome editing, actually, that's when, you know, you just make very precise change in the endogenous genome of that plant. So this is very precise, you know where you are making change, and the variations are very, very predictable. So this is less time consuming, but also if there is no foreign gene integration, actually, in terms of the regulation, they are treated similarly like the conventional products. So, you know, you can also save this regulatory hassle there. So the gene editing, the potentials are real. Now we started already seeing the application of gene edited. I already mentioned that the CRISPR-Cas based gene editing, it got the Nobel Prize in chemistry. Jennifer Doudna and and Emmanuel, they got the Nobel Prize in 2020. And so you can use gene editing starting from the functional genomics, like, if you're a breeder, you have QTLs, you want to validate your candidate genes, then you can use the gene editing for that. You can use the gene editing for nutritional enhancement, for the yield improvement, for biotic stress resistance, like against the pathogens, bacteria, virus, fungi. You can also use it for abiotic stress tolerance, and as well as the herbicide tolerance as well. So far, gene editing has been applied in more than 40 crop species across more than 25 countries, almost targeting these traits. And one thing which is very nice about this is, like, when it was the GMOs, actually GMOs were more dominated by private sector. But when it came to the to gene editing, actually it is more in the public sector, as you can see. So the down bar is the public sector, and this is the private companies, the big. So the gene editing is more by the public research institution or even the small startup companies. And there are quite a lot in the pipeline, but so far there are only few products which has reached to the market, because is a quite new technology. So one of the product which is already in the market is actually the high oleic soybean oil, and this is already in the market in US. The second product which is also already in the market is these tomatoes, and they are GABA tomatoes, like rich in gamma aminobutyric acid. And so these tomatoes can be used for fighting the high blood pressure and also insomnia. Then there is this non-browning mushroom, which has been approved in US and Canada for the release. There is also this rice, which has been approved. This is the blight-resistant rice, which has been approved by USDA and regulators in Colombia as non-GM. Argentina and Brazil and Colombia has recently approved drought-tolerant soybean as non-GM. And in last year in November, actually, Kenya also approved the gene editing maize resistant to maize lethal necrosis as non-GM. And so they will be going to the field trials very soon in Kenya. So the potential for gene editing is really high because the technology is relatively accessible, affordable, and even the public and the small private sector can avail that. It can be used to prioritize in the African countries, like for the crops and the traits, the farmers and the consumers are interested there. So you can see there is quite a lot going on in Africa. I will little bit touch about that later. So it can address the important SDG goals, like particularly the food and nutrition security and enhancing livelihoods, also the mitigating climate change and supporting more sustainable agriculture system. But what we need right now, where actually a good progress has already been done, is an enabling environment in Africa with a science-based regulatory guideline for the release and the adoption of these products. And we are seeing several countries have actually already developed the guideline, and others are in process. But at some point, we also need to see how these guidelines can be harmonized among different countries. So many researchers in Africa are actually exploring the potential of gene editing in developing crop varieties for better and more sustainable African agriculture. So I can give some examples, like in Egypt, they are developing the Salwan mutant, That means they have knocked out the SAL gene for the drought-tolerant wheat using the CRISPR-Cas genome editing. In Ethiopia, they are working on teff, which is actually staple food crop in Ethiopia, for making them semi-dwarf and the lodging resistant. Ethiopia is also working on improving the oil qualities of the Ethiopian mustard. In Uganda, the researchers are working on cassava. They're trying to edit cassava for synchronizing the flowering. In Kenya, actually, there are several projects going on. One of them is the disease-resistant banana, which I will actually talk more in detail. There is also MLN-resistant maize. There is a striga-tolerant sorghum. And then there is a project on the virus-resistant potato. In South Africa, also, they are working on cassava for developing the virus resistant. In Nigeria, also working on cassava and for disease resistance and enhancing micronutrients. In Ghana, actually, there is a project on sweet potato where they are trying to improve the yield and the beta carotene. And in Burkina Faso, there is a discussion going on for the gene edited rice resistant for the bacterial blight disease. So now I will maybe talk about more detail to give you example from banana, how this technology can be applied. And as you all know, banana is one of the major staple food crop grown in more than 136 countries with an annual production of 163 million tons. But when it comes to the Africa, actually 1/3 of the global banana produce is produced in Africa. Food security and the cash crop. And in some of the African countries like Uganda and Rwanda, the consumption of bananas are pretty high. It can reach up to like 191 kilogram per year per person, which is actually very high. And they can eat the varieties like matoke or in this form as well. But still, even though banana is so important, actually there is a huge yield gap in banana. So this is the cooking type of banana called matoke. So the potential, the orange bar shows the potential and the purple bar shows the current yield. And you can see there is a huge gap. And this is the plant. This one is plantain and is the same case for the plantain. And basically this yield gap is because the banana production is constrained by several pests and pathogens. There are fungal diseases like black sigatoka, fusarium wilt. There is a bacterial wilt disease. There are viral diseases like banana bunchy top virus, the banana streak virus, and then there are pests like nematodes and weevils. The major problem is that, several of these pests and pathogens are they are coexisting. That means they are present in the same field. So many times the farmer is not only tackling one problem, but is more than one problem in the same field. As you can see from this figure, where the multiple dot means the multiple pest pathogen present there. So at IETA, actually, we have a genetic improvement program for banana, and our focus is the disease resistance. And so we use all the different tools available for us, because what we are trying to do is we are trying to develop the improved banana varieties with a broad spectrum resistance to various diseases and pests. So that means more than one disease we are targeting. We start with the selection of the resistant varieties if they are already available. We also have the conventional breeding program, and then we have the biotechnology where we are actually using the genetic modification, that's the transgenic, and as well as the gene editing, whatever is needed. And the way we decide on that one is so we have very strong team for the conventional breeding, but as I explained before that, if one of the parent doesn't have the trait which you are interested in, then actually is not possible to cross. But the advantage with the conventional breeding is it is low cost and it doesn't have any regulatory issues. Then we go to the genetic modification. If there is the trait is not available in the germplasm, then we actually were using the transgenic approach where we bring in from outside of the banana Musa species. And this is a medium time projects, but the cost is high because there is also the safety regulation is required here. We also use the gene editing where we only tweak the banana genome because we are actually making the precise changes in the endogenous gene of banana without bringing any gene from outside of that species. So this is low to medium cost, and there should not be any regulatory issue for these products to take them to the market, but also regulation depends on the country policies. So we started the gene editing work at IITA long back in 2015, because once you start working on any crop, actually first you need to establish the system, and then before you go on the trait. And so right now we are working on bacterial wilt, and that's and all, as well as the fusarium wilt. And then our target is that by end of this year we will start a field trial, particularly for the bacterial wilt resistant gene edited. So in order to establish the gene edited system for banana, we used a visual marker gene. So that, when we are editing, we can see that. And the marker gene we have used is called a phytoin desaturase. So this is the PDS gene, which is actually present in the carotene biosynthesis pathway. And then these PDS genes are actually involved in the chloroplast development. So when the PDS gene is functional, the plants are green, but by editing, if we create the mutation in these PDS and make them non-functional, then actually the plants are albino. So we use this system to establish the gene editing for both banana as well as the plantain. So what we did was we designed the guide RNA targeting the PDS gene, and then we delivered the CRISPR-Cas reagents into the cell suspension. This cell suspension has millions of cells here, and then each cell has the potency to develop into the complete plantlet. So when we regenerated, you can see our mutants are albino here. And we further actually confirmed the mutation by doing the target sequencing, and we saw some small deletions or in these plants, so that we know that the PDS has become non-functional. That's why the plants are albino. After establishing the system, actually, then we targeted on the traits, and our first trait is the banana streak virus. So before we say, let me tell you a little bit on banana. Banana is triploid, and the bananas came from two wild progenitor, the cultivated varieties. They came from two wild progenitor, the Musa acuminata, which is a diploid and it has AA genome, and then the Musa balbisiana, which is also diploid and it has BB genome. So all the cultivated banana varieties are triploid. They can be triple A's, they can be AAB, or they can be ABB as well. And so this banana streak virus is a double-stranded DNA Badna virus, like you see here. And so this is this virus, it has a very peculiar thing because it's a Badna virus. All Badna viruses has that characteristic that they integrate into the host genome. And the banana streak virus, actually BSV, gets integrated into the B genome of the banana. So basically B genome is in plantains, because plantain has AAB. So one of the genome is B. So this streak virus, it is a circular monopeptide genome, and it has 3 open reading frame, open reading frame 1, 2, and then the red one is the 3. So open reading frame 1 and 2 are small open reading frames, and basically they are involved in virion assembly. But open reading frame 3, it is a polyprotein. That means it requires a cleavage, a post-translationally, and in order to make the functional protein, and it encodes all the important proteins required for the viruses, like movement protein, code protein, aspartic protease, reverse transcriptase, and RNase H. And then this the cleavage on the polyprotein actually happens at the aspartic protease. And when it integrates into the B genome, it actually integrates in a multiple direct or indirect tandem repeats, but it only integrates at a single locus in the B genome on the chromosome 1. And so when this one is integrated in the plant genome, basically the plant, it just sits as a part of the plant genome. That's why it's also called endogenous, and that's why it's called EBSV. And plant doesn't show any symptoms. But when plant consider stress, that stress can be the environmental stress as the change in the temperature or drought, or the challenge, or the stress can be actually the tissue culture, because the banana is a vegetatively propagated crop, and so it's multiplied through tissue culture. Or even during the breeding, actually this gets activated, and it gets activated into this episomal form, and that's when the plant shows this symptom. And when this plant is showing symptoms, the yield reduces drastically. But this virus has become more of a challenge into the breeding program, and the breeders cannot use the diploid Musa balbisiana in their breeding program, or any parent which has the B genome, because of this virus problem. And it's also a problem for the germplasm movement. So you cannot actually move plantain or the hybrid plantains from one country to another because of the presence of this endogenous integrated banana streak. I still remember that long back, I wanted to actually get the plantains actually from into the Uganda from Nigeria, and I couldn't get any approval from the regulator to bring any plantain from Nigeria because of this problem. So we decided to actually edit this viral genome, which was integrated to make it inactivated so that it doesn't activate into this form of virus. So the theory which I used was like, okay, if I can make all these open reading frame of the virus non-functional, so that the virus virion assembly cannot happen. And what I tried to do is, like, we created a mutation in the open reading frame 1 and 2, and also in the 3 at the aspartic protease. So my idea was that if I make the aspartic protease non-functional, then actually I will make the whole open reading frame 3 non-functional, because the cleavage of the polyprotein will not happen in that case. So what we did was we worked on a plantain which is commonly grown in Uganda called Gonja manjaya, which has AAB genome. And then so we picked the plantain which was not showing any symptom. And then we characterized that plantain to see what is happening. So we saw in the plantain actually this integration of the banana streak virus. We did a lot of molecular characterization in order to come to this conclusion that, there was multiple copies of the banana streak virus has been integrated. After that, we designed the guide RNA, and we did 3 guide RNA targeting each open reading frame 3. And then we have assembled all the CRISPR-Cas reagents into the one plasmid, and then we we delivered those reagents into the cell suspension of plantain, which is Gonja manjaya, and we regenerated the complete plantlets. After regenerating the complete plantlets, again, we did some analysis to actually check the mutation, the target mutation. So these are our control wild type plants which were not edited, and these are the edited plants. Phenotypically, there was no difference. They were looking the similar. So what we did the target sequencing, and after doing the target sequencing, we saw the deletion. Because the open reading frame 1 and 2, they were small, and then if there was a simultaneously deletion, then we were expecting about 198 base pair deletion, which we saw. And then in case of this for the open reading frame 3, we saw the small deletions as well. So after seeing this, confirming the mutations, actually, then we have evaluated these plants for the symptom. So we put these plants in the glass house, and then we have actually mimic the drought. So that means we didn't give them water for some time. And so this is our control plant. You see the beautiful streaks, they started showing the symptom, but our mutants actually didn't show any symptom. But few mutants, these 2 mutants actually also showed the mild symptom. So we were very curious to see what happened. So these, our 75% mutants, which didn't show any symptom, actually they have the mutation in all the 3 open reading frame. And so which has actually confirmed our hypothesis that, you know, open reading frame 3 is very crucial in symptom development. And then because it's a polyprotein, and then the mutation was at aspartic protease, so that means it has made the whole open reading frame 3 non-functional. So now we have a proof of concept, and we are now trying to actually also added some of the parents which are used in the plantain breeding. Let me quickly give you an example from the banana Xanthomonas wilt, which is a bacterial disease. It is a very serious problem in the Great Lakes region in the East Africa. And the losses can be somewhere between 40 to 80% because of this disease. But the major problem is once the disease is there in the field, actually the whole complete field gets wiped off in very quickly in a few months time. And then there is no other remedy than to uproot that all the plants and then leave the field fallow for some time before the farmers can actually replant that one. And this disease actually it spreads through the insect or also the infected planting material and through the contaminated farming tools. All the varieties, the cultivated varieties are susceptible to this disease. But what we noticed that, you know, the wild type banana like Musa balbisiana was found to be resistant to the disease. And overall economic losses were estimated between 2 to 8 billion over a decade because of this disease. So we are trying to understand the resistance in the Musa balbisiana so that we can actually develop the resistant varieties. And this is the picture from Rwanda, where, you know, the farmers have completely uprooted the whole field because of this problem of the banana bacterial wilt disease. So we did comparatively transcriptome analysis, RNA-seq, and we compared the Musa balbisiana, which is a resistant diploid, with a susceptible variety. And based on the RNA-seq work, we actually identified a lot of differentially expressed genes. And then we mapped those genes, DGs, against the biotic stress pathway. And that's how we have identified a lot of candidate genes which we can use them as a target in the gene editing. And we have further validated those genes as using the qRT-PCR to confirm before we wanted to use them into the through gene editing. So right now, what we are trying to do is, like, we have now the information from this Musa balbisiana that which are the genes which are involved in development of the disease or providing resistance against the diseases. And then we are actually transferring that information or knowledge from the Musa balbisiana to our susceptible varieties. So we are not actually transferring any gene from here to here, but we are using that information. So we are using either the overexpression of defense genes through the CRISPR activation. So that means we are overexpressing the endogenous genes in our susceptible varieties using CRISPR activation, or we are knocking out the susceptibility genes. So that means we are making the susceptible genes non-functional in the susceptible varieties. So that's how, so we are using CRISPR-Cas for knocking out, and we are using CRISPR activation for upregulation of the endogenous genes. So for the upregulation of the defense gene, we have tried these different genes. We have tried, like, resistance gene, we have tried PR1, and we have also tried some of the receptor kinases like WOC2, WOC5, and we have tried these antimicrobial gene. All these genes we have identified based on our transcriptomic work. And then what we did, we used the CRISPR activation technology to actually simultaneously activate 3 of these ones. So we developed 6 different constructs, and then we have tested them. And then we have, so we developed the plants. And then after developing the plants, actually, we did the qRT-PCR to check the overexpression of these genes, but we also did the glasshouse assays to check the resistance. So some of these events, like, you see the 11, they have shown very nice high complete resistance to the disease in comparison to the control, which were completely wilted. We also actually tested the knockout of several susceptibility genes. We are testing a lot of genes like SWEET14, MLO, early Notulin, PAB, BS2, ADH1, and DMR6. So I will give you example from the DMR6. So DMR6, downy mildew resistant 6, this is a susceptibility gene, and it is actually activated during the pathogen infection. So when the bacterial infection happens, then, this gene can go upregulated, and then in return, it suppresses the plant immunity, and which we have actually tested. So this gene actually is very well characterized in tomato and Arabidopsis. So we have also done the phylogenetic tree against this, the whole gene family, and we identified an ortholog of the DMR6. And when we actually tested it, so when we have infected our susceptible variety, we saw 60-fold upregulation of the Musa DMR6 in that one, whereas our control and the Musa balbisiana, which is the resistant variety, we didn't see any upregulation of that. So that has also confirmed that the gene which we have identified to knock out is the correct gene. And then after that, we have designed the guide RNA, and we have knocked out this gene. We have regenerated the plantlets, the mutants, the DMR6 mutant, and then in the mutants, we actually confirm the targeted mutation by sequencing. And after that, we have tested them for the resistance to the banana xanthomonas wilt. We have developed actually very robust in vitro assay. And so initially we tested with this in vitro assay, and in this one, you can see the last one is the control, the complete wilting of the plant, whereas our mutant showed enhanced resistance to the disease. We further tested that in the glasshouse, And using the potted plant, we have artificially challenged our potted plants with the pathogen, bacterial pathogen. This is the control plant which has completely wilted, and whereas this is our mutant which has not shown any symptom of the disease. We also checked that knocking out this gene has no negative impact on the plant growth. So we have actually we took the plant height, pseudostem girth, the leaf area, and we didn't find any negative impact of this disease. So this is published paper. If you want to read more, you can go back to the Plant Biotech Journal, and then you can read the full paper there. So these was the examples I was giving from banana, but we have also actually established the gene editing system for yam, the Dioscorea rotundata. And you know that the one of the requirement for doing the gene editing that you should have the reference genome of that variety, but also you should have you should also have the regeneration system or the transformation system. And then we have established the gene editing system because for yam, there is the reference genome available for both Dioscorea rotundata and alata. And we again use the same visual marker, the PDS, but this time our guide RNAs were designed specific to the genome of yam. And then we have already now the yam, the system established. Now we would like to use this system for developing the resistant to the viruses and the anthracnose disease. As you know that the gene editing is new, so there is actually a need that, we have to communicate for the public awareness, but also the capacity need to be built. So we are heavily involved actually in the capacity development of the young researchers. So we are training PhD students in our lab. So far, we have trained 4 PhD students who have been graduated. We also do short-term training in the lab. And recently, actually, the IITA in partnership with UC Davis and the African Plant Breeders Academy, we are actually training 11 students through the CRISPR course. And so this is the first batch of a student who started recently in January. And this course, we will run this course for like about 5 years. And our intention is to train about 80 to 100 breeders in applying the gene editing in the crop of their interest for their and also the traits their country is interested in. And we will also provide to these students some mentorship so that when they are back to their institute, actually they can apply this gene editing in their host institution as well. And we are working very closely with Alliance for Science actually also for communicating. So we are training a lot of people actually how to communicate the science to the public as well as to the regulators as well. So okay, that's the almost end of my talk. And so the key take-home messages are that gene editing tools are becoming the popular molecular tools of choice for not only for crop improvement, but as well as for the functional genomics. And among all different tools, CRISPR has become actually the most popular genome engineering approach because it is simple, is efficient, very specific. You can do multiplex and easy to adapt. What you need is the reference genome, the plant transformation protocol, some bioinformatics tool, and the regulatory framework in order to actually adapt the CRISPR-Cas into your lab. The gene-edited crops with no foreign gene integration are treated similar to the conventional crops in several countries. I gave you that example. The application of CRISPR-Cas based gene editing has been actually demonstrated in several crops, including banana. And building capacity, creating a critical mass is very important. And that's why we are involved in training a lot of young African researchers in applying gene editing. So in the end, I would like to acknowledge my team at IITA, both the plant transformation as well as the bioinformatics, and then my partners from UC Davis, Iowa State University, Cornell Alliance for Science, and also the financial support from USAID, the CGIAR research program, NSF, and BMGF. So thank you very much. I'll be happy to take any question. Thank you.
Thank you so much. It was a good long 1 hour, but very easy to follow, comprehensive. The work that you're doing, a very good presentation. Any questions, comments? So far, there's nothing in the chat, but if you, Benjamin Karihari, you just introduce yourself, where you come from, which institution you are with, and then your question.
Okay. Thank you very much. I'm Benjamin Kakari. I work with University for Development Studies in Tamale, Ghana. I have expertise in CRISPR-Cas, precisely soybean. And since I arrived in Ghana upon my PhD training, I have been searching for such opportunity to. I have read a couple of papers from the presenter, and thank you very much for transferring your knowledge and skill to the younger generation in Africa. As you mentioned earlier, African Union has initiated some consultative dialogue on CRISPR-Cas, of which Ghana is included. But I would like to ask, are there some possible other avenues that one can tap for funding? Because most of the facilities that we have, we need to start everything afresh. And it calls for huge investment, which currently I don't have, or my institution hasn't provided. Are there some possible through your network that we can also probably build on what we learned or what we applied in soybean, and then other crops that are of importance to us in Ghana? Thank you.
Dean, you want me to answer?
Yes, yes, yeah, we can respond.
Okay, so I think that when it comes to the funding, we all are facing a big challenge of the funding right now for the gene editing. Because that's why you see, we have established the system for yam, but we are not doing anything for last several years because we don't have funds to move on that one. Because there are still very few donors who are supporting gene editing. So I don't know what I can say. I can only say that, you have to maybe develop a project proposal and discuss with some of the donors who are interested in your crop and in the country. And if you need my help in terms of technical help, I will say, for the proposal or something, please feel free to approach me through email, and we can discuss that. But apart from approaching some of the donors like BMGF, USAID, maybe some like African Union, I don't know where else you can tap on the opportunity. Maybe I don't know if your university has some funding as well.
Thank you very much.
Thank you. Let me go to there's a comment. This is a comment. Thank you very much for your nice presentation and for sharing the information about gene editing. Would you share us the record? We will share with all the participants. I think you are registering. We got your emails. So we will share the presentation and the record. And then I am Lillian Noela Wesonga, a PhD plant breeding student at WACI. I had the privilege to be trained by Dr. Tripathi's team on gene editing as a graduate trainee. I was sponsored by the Alliance for Science. It was such an excellent learning opportunity. Thank you so much for the efforts that your team. This is just a comment. Question from Marilyn Christian. silicon has been used as a fertilizer amendment to improve nutrient uptake and a range of biotic and abiotic stresses, including plant diseases. Recently, silicon genes have been identified for silicon uptake, and its incorporation has been shown to improve stress tolerance. Would you see future research in stress tolerance breeding, including the use of silicon genes in CRISPR-Cas breeding? Then I think before, maybe let me take the second one from Mary. Leena, thank you, Dr. Leena, for your presentation. I would like some clarifications about the organism targeted to be edited, the pathogen, the host plant. If you edit the virus, could the plant not be reinfected? So I think those are the 2 comments you can respond.
Yeah, thanks, Dean. Let me start with the second question first. For the gene editing, right now we are targeting plants. We are not targeting pathogen. And so because we have more information, and then regarding so that was her first question. Her second question is the virus. So we have still not targeted the virus. What we have targeted is the viral genome, which has been integrated into the banana. That's what we targeted in terms of the BSV. And as you remember, I said that this virus integrates only on one specific loci in the plant, on the B genome of chromosome 1 of the plant. And so, what we have done is we have not removed the whole viral sequence from them. We have only created some knockouts, like some deletions in the viral sequence, so that the viral sequence cannot assemble into the activated virus again. So in other words, we have inactivated the virus. So in that case, there is no open space on that loci to the another virus sequence to come and integrate. So that's why there is no chances of reinfection, but we have not tested that in the field yet. Okay, so now going back to the silicon question, basically, editing it looks very simple when you talk, but it's not that simple. So that when you want to have a target, you need to understand the whole pathway. So if you want to if you want to work on the stress tolerance targeting the silicon, you need to actually understand the whole pathway. And you also need to understand what you want to do with that gene, whether you want to overexpress that gene or you want to knock out that gene. So you need to complete full information of that pathway before you can decide the target to edit using CRISPR-Cas. Because you remember I showed before we started working on the vector wilt, we spent actually almost 2 years to understand the pathway, the biotic stress pathway in banana before we chose the target genes. So I hope that answered your question.
Okay. The other one is just a comment from Irene. Insightful presentation. Thank you so much for the opportunity to learn a lot from Dr. Tripathi. Then this one is not named. I appreciate it for your nice presentation. What is the prerequisite to do gene editing?
So the prerequisite, as I mentioned, is actually definitely you need a lab. You need the reference genome for your crop. You need the plant transformation system for your crop. And then you should know some bioinformatics before you can start editing your crop.
Okay, I see Mary has raised her hand. Mary, you can go ahead.
Thank you. Can you hear me, please?
Yes, I can.
Thank you so much, Dr. Leena. It's always a pleasure to hear from you. I just thank you for clarifying that is the plant, because I asked a question. I was one that asked the question. You said the plant is the one that you target for editing. Now, I want to know, because it sounds to me like, yes, you are refining or maybe changing the plant's sites where the virus comes to bind. The virus wants to come in to penetrate into the plant, and then you kind of modify those proteins at the surface level of the cell for the plants so that the virus does not have the ability to bind. Is that what is happening here? Or what?
No.
Because I know about
no,
how
we
the
are not
disease
doing
is.
that.
Okay.
We are not doing that.
Okay.
So in this case, the banana streak viruses are the Badna viruses. So these Badna viruses, when they infect, so we are not doing anything with the infection. So when the virus infect the plant, actually the viral genome goes and integrate into the banana genome.
Okay. Okay.
So what we are targeting is the viral genome, which is already integrated into the plant genome. So we are trying. So what happens that when virus infect, actually the plant doesn't get sick. The plant only gets sick when this integrated virus actually become activated into the episomal form. So that's the cycle which we are breaking. So that even though the integrated virus is there, we are making the integrated virus. We are in a layman language. If I say I'm shredding the viral sequence which is integrated, so that it cannot assemble back into the virion, the active viral sequence.
Okay, so
okay, thank you so much. Yeah,
we're getting more comments.
Maybe this is a published paper. I think you can also read. This is published in the communications, the Nature communication paper, and you can read it. Maybe you will get more information from there.
Okay,
thank you.
This is a comment from Uma. Yeah, we will consider both registered, because some were dropping. We got to about 90 participants. I think that was the highest so far, which was good. So we'll share because as you were joining, there was a registration process, so we use that. Then from Adekemi Stanley, thank you for your nice presentation. I would like to learn more on CRISPR. Are there training opportunities? I think you've highlighted. Maybe if you want to say one or two words, because I'm sure you highlighted about the communication, learning, training opportunities.
Yeah, so the training opportunity, one is the CRISPR course I'm talking about. So, we have the first batch of students, which are, so we will take one batch every year. And these are the students from 7 different countries, which are currently under training. So the second batch will come next year. So when towards the end of this year, we will again advertise. You can check on the IITA website or even the UC Berkeley website. We open the call and then you have to apply. This is a competitive. So, not because this year we got 56 application and we selected 11. So that's how it will be. Then another one is that, if you can also arrange through your institution, if your institution is willing to cover the cost for the training and willing to send you to my lab, that's one opportunity always. We always accept the students through the support from your own institution coming to my lab.
Okay, thank you. Then this one is from Masoud Saleh. Thank you, Dr. Tripathi, for and the organizers for the good presentation. We hope that the program will continue and make APBA a good platform for knowledge transfer, especially for young scientists. So like I said, we will have these webinars the first Wednesday of every month. So the next one will be on 5 April 2023. And we have a Facebook page, we have a Twitter and LinkedIn. Please follow them so that at least we'll be able to get this. Then my name is, I am Drabo Inousa, a pure millet breeder at Simit, Senegal. Thank you for the great presentation. How to communicate so that the regulation makes difference between GM edited and other kind of GM. I think it's a question. How do you communicate so that the regulation makes a difference? How do we,
Yeah, so basically, we are working very closely with the regulators in the countries which are the countries where we have the target. And I can give you the example from Kenya, because like in 2015, when I wanted to start this, I approached the NBA. And then, so when we started our first training, we started in 2017 for communicating the science behind gene editing. And several of the participants actually we invited from the regulator. So that when this is a new science, even regulators needs to understand. Then also when there are NBA also organized many times a workshop with their board members, NBA as well as cafes, and then they invited people like me and other scientists to come and explain and what is the difference between gene edited crop and GMO, so that they understand so this is not like a one time talking to them. This is like a regular discussion in the countries you are in, with the scientists, the communication experts, and the regulator. These are the three key, and then the discussion keep on going before actually everything works out well, that they understand the difference between the gene edited crop and GMO and develop the guidelines accordingly.
Okay, thank you. The next one is from Derek Mudadirwa. Thank you for your nice presentation. My name is Derek. I am a Pan-African University student. My question is, when CRISPR-Cas is used in sexually propagated crops, are the targeted traits inherited for several generations, or it needs to be repeated often?
That's a very good question. Actually, it is inherited. So once you make the mutation, that's inheritable. So you don't need to create again. And not only in the sexually propagated, but as well as the vegetatively propagated crops as well. And we have already tested that in bananas. When we were doing this banana streak virus, actually, we have propagated our bananas for 6 different generations. And then we tested the progeny, and then the same mutation, not even with any change, we found them in the progenies as well.
This is just a comment from Percy. This has immensely impacted my knowledge on gene editing and the use of CRISPR. Thank you, Dr. Tripathi and APBA for the opportunity. And then Ali, it's also a comment, OG Ali, legume breeder from Tunisia. Thank you very much for the great presentation. I don't see any hands. I don't see any more comments. I want to thank you so much for the long 1 hour, 15 minutes. It was worth it. You could see from the comments, many people, it's a new technology, easy to use and cheaper, and also acceptable, because some parts of it, they don't require that regulation. So it's an opportunity for breeders. So thank you so much. And then to the, so in this presentation partly is also featured in our newsletter. So please look for our newsletter. We'll share the newsletter as well, so that at least you read through. Like I said, the next webinar is on the 5th of April, 2023. Every first Wednesday of every month, we're going to have a webinar. So thank you so much, Dr. Tripathi, for your time. I know in East Africa, it's almost 4 p.m. Here in Nigeria, it's still 2 o'clock, so it's good. And so thank you so much for your time, accepting to do this at a very short notice. Thank you so much.
Thanks, Dean, for organizing it.
All right, that marks the end of this webinar. Thank you. Bye-bye.
Bye. Bye, everyone.
Bye.
Bye. Bye. Thank you so much, Ma.
Bye.
You're welcome.
Bye.